I would not be here, I would not be a scientist without my mentors, Lucas and Scott. Scott was my postdoctoral mentor in the US, and Lucas was my mentor during my PhD. And when I started my young research group here in Innsbruck, and over time, Lucas and I have become dear friends, and Lucas is always there when you need advice and when you need help. And what I learned from both Scott and Lucas is that for doing science, and not just being a scientist, doing science is something different. For doing science, you need to embrace at least two things, and probably three. You need to be very precise in observation. You need to pay attention to details, and where you also, of course, have to do hard work. But I also think that these two abilities are not enough if you want to be a really good scientist. I think at the end of the day, you also need passion and heart to pursue your dreams. And God, I'm getting sentimental. It has to be over. Over the years, I was lucky to work with a large crowd of people and with excellent students who have helped in shaping my ideas, and I wouldn't be here without their help. So, but enough of the sentimentality, let's talk science. To illustrate what molecular membrane biology means, I'm going to use a lattice light sheet microscopy movie of single-cell organisms that we like to study in the lab. These are yeast cells that express a nutrient transporter. And when the movie starts, they experience a change in the extracellular environment. One amino acid is changed, and what they do is that they start to downregulate the amino acid transporter that is responsible for the uptake of these individual amino acids. And they do that within 20 minutes. Within 20 minutes, every single one of these amino acid transporters has been cleared from the surface of these organisms. What is a membrane built of? A membrane is built of two major components. It is built of lipids, and it is built of membrane proteins. Proteins are actually rather static entities. Lipids, on the other hand, are very different. The lipid diversity of a membrane is encoded by the range of the genetic possibilities that are given within an organism. And how membrane proteins and lipids interact with each other and form a functional membrane is something that I and my team would very much like to understand. So if you want to understand something, what you should first always do is to make a list of the inventory. What are the components of the system that you are going to study? And we like to study membranes in two model systems. One is a single-cell organism, it's Baker's yeast, Saccharomyces cerevisiae. And in Saccharomyces cerevisiae, we know that the genome encodes for 1,200 transmembrane proteins, 26 lipid classes, and over 100 different lipid species. In human cells, this is a cell line that we like to use in the lab. They're called RP1 cells. We know that the genome can encode for 6,000 transmembrane proteins and more than 1,000 different lipid species. And in theory, 13,000 different lipid species are possible, but nobody has ever detected those. Regardless, you can see that the combinatorics that are possible between membrane proteins and lipids are huge. So how do cells define what is in a membrane and what is not in a membrane? How to decide which components are properly adjusted in the right spot and which are not? Looks like a simple question if you consider the classical Singer-Nichols model, but the Singer-Nichols model fails because it, of course, assumed that membrane proteins are widely dispersed throughout a sea of lipids. Instead, if you look at anatomy, and also anatomy can be part of a research program, you can see that these vesicles are filled with many, many different proteins. This is a model of a synaptic vesicle that is 40 nanometers in diameter. And we know that this synaptic vesicle consists of 160 different proteins, 80 of which are integral membrane proteins, and 7,000 different phospholipids. And the protein to lipid mass ratio is actually 2. So membranes are packed with proteins. Another illustration is this illustration here that I have taken from a website that is called Cellscape. Here you see the endoplasmic reticulum touching the outer mitochondrial membrane. And you can only appreciate, or you can start to appreciate, how many different membrane proteins are embedded in just this very small section of a membrane. So how does a cell figure out which proteins to remove, which proteins to insert, which proteins do no longer work, which proteins are working? How do cells do that? We want to understand that. And so this basically opens up the major questions for membrane quality control that we are interested in my lab. What we would like to understand is how selective membrane protein degradation pathways control, on the one hand, nutrient uptake, in particular the uptake of amino acids, and how they also control lipid metabolism, and how these two processes control cell growth or phases of quiescence when cells do no longer grow. How do these processes intercalate and interact with each other? Right. So what we do know is that membrane proteins have been considered to be degraded by two exemplary pathways. One of them operates at the ER, the endoplasmic reticulum, which is called ER-associated degradation. The ER, if you like, is the birthplace of the vast majority of all membrane proteins. And immediately, if something goes wrong for the biogenesis of membrane proteins, the ER-associated degradation pathway will find these defective membrane proteins, ubiquitinate them, extract them into the cytosol, and target them for proteasomal degradation. Once membrane proteins are properly folded, they can exit the ER. And once they exit the ER, they can go to the Golgi, to the plasma membrane, to endosomes, to multivesicular bodies, or to lysosomes. And then these membrane proteins are typically degraded in lysosomes and are ubiquitinated by mechanisms that are not really well understood. But it was always thought that these ubiquitinated membrane proteins are sorted through a pathway that is referred to as the multivesicular body pathway into lysosomes for degradation. So for the first bit, I will talk about this escort pathway, this lysosomal degradation pathway, in relation to how membrane proteins and nutrient transporters are degraded in cells. So we wanted to understand how nutrient transporters are degraded because actually nobody has bothered studying that. Although it is very clear that nutrient transporters that are required for the uptake of amino acids or sugars are absolutely essential for life on Earth as we speak. And of course also have important roles in the control of cell growth, metabolic reactions, and so on and so forth. But nobody really understood how these nutrient transporters were regulated. And to start this in human cells or in to study this in humans is a daunting task because the human genome expresses 14 different glucose transporters, 66 different amino acid transporters, and recent counts that were actually published last week estimate that the human body contains 36, the male human body contains 36 trillion cells, the female human body 28 trillion cells, and roughly 400 different cell types. So trying to understand nutrient uptake in such a system from scratch is nearly impossible. Therefore, we decided to go back to budding yeast because there we only had to deal with 6 glucose transporter, only 24 amino acid transporter, and the entire organism is built from a single cell. So things are much simpler in yeast. So what we first did is, again, to chart the inventory of these nutrient transporters, and we found that under normal growth conditions in the lab, roughly 50 transporters were detected at the plasma membrane, including 7 different amino acid transporters and 6 different sugar transporters. And then we observed that as cells were entering quiescence, in this case by taking away amino acids, they would begin to downregulate a defined subset of their amino acid transporters. So we figured out that under these conditions, when they entered quiescence, they downregulated 4 amino acid transporters, 4 sugar transporters, and 6 other plasma membrane proteins. But 35 other plasma membrane proteins were not regulated at all. 1 unspecific amino acid transporter and 1 ammonium transporter were even upregulated. So when we knew that, we already knew something very specific must be going on because the cells that enter quiescence select very specifically a certain subset of nutrient transporters. And we also could show that this process is essential for the entry into quiescence. And once we knew that, we also could devise a strategy, Lucas called it nicely a genetic tactic, to screen and to identify genes that were essential for the downregulation of these nutrient transporters. And I'm not going into these details here, but at the end of the screen where we scored every single viable yeast mutant, we identified 128 genes that were absolutely required for the downregulation of these nutrient transporters. And from these lists and additional experiments, we could build a model in which we could describe in budding yeast for the first time that nutrients control the so-called integrated stress response. This is a pathway that controls directly the translational activity of ribosomes. By controlling this translational activity, this pathway allows the translation of a so-called alpha-arrestin protein. And this alpha-arrestin protein acts as a ubiquitin ligase adapter, which then targets this ubiquitin ligase, RSP5, to the 4 amino acid transporters that are downregulated. So in yeast, we had figured out how that pathway works. In human cells, this was still entirely unclear, yet we were confident at that point that we could also solve or begin to solve this problem in human cells because all the components that control the amino acid transporters in yeast are highly conserved in human cells as well. So we have this integrated stress response in human cells, we have alpha-arrestins in human cells, and we of course have ubiquitin ligases in human cells. Right, so once these nutrient transporters arrive on endosomes and are ubiquitinated, they cannot just enter like that into lysosomes. They have to be sorted through the multivesicular body pathway into these intraluminal vesicles. And this process is catalyzed by the escort machinery. And this approach is spearheaded currently by Asta Purva and previously had been carried out by Simon Sprenger, Simona Miliano, Manuel Alonso, and we did this work together with John Briggs and Tom Kirchhausen. And what you see here, this is not an artificial rendering, but this is correlative light and electron microscopy where we were able to localize the components of the escort machinery right to the site where they start to deform the membrane and where they start building these intraluminal vesicles. And so our work over many years has then led to a dynamic model in which we could describe how this process occurs. The formation of these intraluminal vesicles and the sorting of these ubiquitinated membrane proteins is a very rapid process. And just to give you a little idea of how this process looks and how these multivesicular bodies look in yeast cells, we have conducted together with Georg Vogel and Michael Hess three-dimensional cryo-EM tomography and used computer power to reconstruct these organelles from yeast cells that were high-pressure frozen. So here you see wild-type multivesicular bodies. This is when the escort machinery can run through their stochastic cycles. It assembles and disassembles. When we only make very slight modifications to the process and throw the machinery off for a few seconds, the process begins to stutter, and these membranes are no longer properly formed. These are hypermorphic mutations. And if we make full loss of function mutations, these multivesicular bodies are no longer formed, and instead, all the membrane proteins will accumulate in these multilamella class E compartments. And over the years, we have made a number of contributions to this field on the endosomal side of the pathway, and we also have helped to understand how the escort machinery helps to maintain plasma membrane homeostasis in response to mechanotensile stress, and how the escort machinery helps to control heterochromatin nuclear envelope attachments. But what always bothered me, despite all these functions of the escort machinery, you can knock out escorts in yeast or in human cells for that matter, and they are fine. You know, how can that be? It already bothered me in Scott's lab. You know, escorts were always, the most important machinery in the universe. Yet they are not essential. If something is important, it should be essential, but it's not. So I wanted to understand why the escort machinery is not essential. There are several possible explanations for that. And you can, try to explain whatever you want, but what you can do is you can do the experiment and try to figure out how can it be that escorts stay alive. So again, when the escort machinery is active, membrane proteins get sorted into the lumen of the vacuole. You build these multivesicular bodies. when you lose the function of the escort machinery, membrane proteins get stuck in these multilamellar class E compartments, yet the cells are fine. So we thought we will use the power of yeast genetics and ask which other pathways are required for the survival of the escort machinery by simply making double mutants in which we knock out an escort component and a component and every other gene in the yeast genome, and then ask which genes are required for the survival of these mutants. And all the suspected candidates which you could come up with explanations were no hits. There were no ERAD components, which is the other known membrane protein degradation system. There was no sign of the unfolded protein response, and also autophagy genes were not activated. And this approach was spearheaded by Oliver Schmidt from the lab, Yannick Weyer, Iduna Schmabel, and Michi Wiederin. But what we found were 3 out of 4 components of the so-called DSC complex. Now, what is the DSC complex? DFC stands for defective in SREBP cleavage. SREBP in human cells is the sterol-responsive binding element. It's a gene that is essential for the control of sterol biosynthesis. Yet, yeast does not have an SRE1 protein or SREBP. Yet this protein complex is called defective in SREBP cleavage. So in yeast, that complex had to do something else. So what we knew is that that complex consisted of 4 transmembrane proteins, one of which is a ubiquitin ligase, a rhomboid pseudoprotease, and for these 2, no function is really clear. And so if you draw the model of membrane proteins or any proteins like this, I could equally well show you an Egyptian tomb image. It's just as informative. It tells you nothing about their function. But since we are in 2023 and AlphaFold is available, you can just send your sequences to AlphaFold and ask AlphaFold to fold your protein complex. And there you go. You get back something that looks like a reasonably folded protein complex. And this is our ubiquitin ligase complex embedded in a membrane. And in the future, we would like to use that as a template to conduct more structure-function analysis on the DSC complex. And I will show you why we think that this is important. So what Olli then, together with Yannick and Michi and Iduna, could show is that this DSC complex is essential for a 3rd membrane protein degradation pathway that had been missed before. So in addition to the escort machinery that functions on endosomes and catalyzes this membrane deformation event that leads to the formation of multivesicular bodies, and in addition to the ERAD system, the EGAD pathway, which at the heart uses this DSC complex, can detect membrane proteins, ubiquitinate these membrane proteins, and extract them from these organelles for proteasomal degradation. That clearly expands the possibilities of regulating the membrane proteome of eukaryotic cells. And we also know, and this is something that Olli is going to follow up, that the first substrate that we identified of this DSC complex is a key regulator of sphingolipid metabolism. So you get here the first connection between the degradation of membrane proteins and lipid metabolism. We are quite interested in understanding what's going on here, because we have now at least 2 examples where these multilamellar compartments form. I've shown you already these class E compartments when the escort machinery is not working. You make these multilamellar compartments where all membrane proteins get stored. I also showed you these novel Golgi-derived compartments. And so I think that maybe these accumulating membrane proteins in growing cells, somehow by a mechanism that is totally unclear, get sorted into these multilamellar compartments to keep the pathways in the growing cells functioning. And so it will be very interesting to figure out how these compartments are built and if they are really either beneficial or detrimental for the cells. And so I hope I could convince you that what we are going to do in the future is studying membrane quality control and its implication for human health. Membrane quality control is just a small part of a much larger membrane proteostasis network where an estimated 2,000 proteins operate. It's almost a tenth of the entire genome that is dedicated to quality control processes. And we will focus on these membrane quality control pathways that result in the formation of these multilamellar organelles, be it class E compartment or these Golgi-derived vesicles. Whether this accumulation of membrane proteins in these compartments protects organelle integrity or damages organelle integrity and therefore causes or prevents cellular damage, which over time might lead to diseases such as neurodegeneration and, in the cases of the amino acid transporters, metabolic diseases. And with that, I'm almost at the end. I've only left to say thank you all. I've mentioned most of the people that we have worked with at the university here. I would like to thank, Lucas, you again, all the members of your department, especially the technicians that I have worked with over time, Brigitta, Yvonne, and Sabine. I would also like to thank Brigitta, André, and Winka for doing all the glass wash, because this is one of the most important jobs at the Institute. And if this is not done well, nothing else works. This is the basis for what we do. And the same actually holds true for Janine Köppen and Gertrude Huber, who keep our secretariat alive and kicking and take away a lot of work from us. And I've mentioned most of the other people that we have collaborated with. And at the end, again, thank you. We already have moved to the 4th floor. We have occupied Klaus Schäfzek's former department. I should send my regards from Klaus. Klaus couldn't make it here today, perhaps because we have taken over his office already. And I'm very much looking forward continuing to working with my people and to continue to work and start work with the people that were formerly members of the Institute for Biological Chemistry and will now be members at the Institute for Molecular Biochemistry. Thank you.